Hello, my name is Jolien, and today I'd like to tell you about why the peripheral immune system matters when looking at immunotherapy, and what people have learned over the past few years by looking beyond the tumor microenvironment. I'll be doing this by summarizing a couple of key studies that were performed in mice as well as in humans. Immunotherapy has been used for the past 10 years in the clinic, and even so, we've not quite understood yet how immunotherapy works. For a long time, people thought immunotherapy controls the tumor by reverting exhausted T cells that are present in the tumor back into an effector phenotype, essentially reinvigorating local T cells. However, a growing body of literature suggests that instead, immunotherapy might work by bringing new, non exhausted effector T cells from the periphery into the tumor, where they replace those exhausted T cells that are present there. I will be summarizing the studies that have led us to believe in this last hypothesis. First, I'll be talking about a study performed by Matt Spitzer and his team that was published in 2017 in the Journal of Cell. They showed in a mouse model of colorectal cancer that peripheral immune cells are critical for effective immunotherapy. In this particular mouse model, immunotherapy is normally capable of controlling the tumor. To investigate what peripheral immune cells were contributing to this, Dr. Spitzer applied a drug to these mice that trapped immune cells in the lymph nodes, essentially making it incapable for them to reach the tumor. What they saw then is that when these mice treated with this immune trafficking drug, when they were treated with anti-PD-1 therapy, there wasn't any tumor control anymore, and as a result, the mice died from cancer. These data suggested that peripheral immune cells were not just important, but in fact required for effective immunotherapy in this mouse model. It's important to note that other studies have since replicated these findings in different mouse models with other tumor types, suggesting that this system holds true across different cancers. So then next the question became, if this is working in mice, then what is happening in humans? And the following years, 2019 and 2020, a series of papers published across Nature Press journals confirmed that humans had a similar system, that peripheral immune cells were critical for establishing effective immunotherapy. By using single-cell RNA-seq, TCR-seq, and flow cytometry, these papers established the following. First, tumor-resident T cell clones, both effector as well as exhausted T cells, were clonally related to cells that were present in the blood before immunotherapy. And in fact, most cells originated from the periphery, like in some patients, almost all of them. That suggested that anti-PD-1 therapy in these patients led to clonal expansion of blood T cells, and that these expanding T cells then migrated to the tumor, where they ultimately took on an exhausted phenotype. To confirm then what was happening in tumors, in T cells that actually originated from the tumor, so that were not present in the blood before PD-1 therapy, they saw that those tumor-resident T cells had an exhausted phenotype, and that upon PD-1 therapy, they maintained that exhausted phenotype and only had limited clonal expansion. So taken together, these studies concluded that immunotherapy controlled tumors by improving tumor infiltration by fresh clones from the blood, rather than reinvigorating or affecting the tumor-resident exhausted T cells. Then the question became, these blood T cells, are they activated in the blood upon PD-1 therapy, or are they coming from another location? Again, a series of papers, first in mice and then in humans, confirmed that blood T cells were first activated in the lymph nodes before trafficking to the tumor. In 2021, two papers came out that showed first what was happening in mice during regular tumor progression. First, they found in the tumor-draining lymph nodes the presence of stem-like T cells. These T cells were able to self-renew in the presence of chronic antigen with the help of local dendritic cells. These stem-like T cells that were maintained throughout tumor progression gave rise to a T cell phenotype that was sort of intermediate between exhausted and effector T cells. Both the stem-like T cells and intermediate exhausted T cells were, during tumor progression later on, found in the tumor. And the author showed that these were the same T cells by doing TCR sequencing and showing that they were clonally related. In the tumor, these intermediate exhausted T cells then ultimately differentiated in exhausted T cells over the course of tumor progression. And finally, to fill in the missing piece, the author showed that these stem-like and intermediate exhausted T cells were present in the blood at some point, suggesting that the draining lymph node resident cells migrated through the blood to the tumor over the course of tumor progression. This was all done in mice, and the next step then was to see if this was happening in humans, which was done by Dr. Spitzer and his team in a paper in Cell published in 2023. They were able to confirm all of this was happening in humans as well, but added the part that upon anti-PD-L1 therapy, it was particularly having an effect in the draining lymph nodes. PD-L1 therapy colloquialized the stem-like T cells with the dendritic cells and resulted in increased proliferation of the stem-like T cells, which then led to an increase in production of these intermediate exhausted T cells. To summarize, the body of literature that I just presented suggests that immunotherapy works by bringing new non-exhausted T cells to the tumor microenvironment rather than reinvigorating exhausted T cells into effector T cells in the tumor itself. Then perhaps it is not too surprising that we see that several immune features after one cycle of immunotherapy are associated with improved clinical outcomes. Several of the Nature Press papers showed that improved overall survival was associated with increased T cell clonal turnover in the periphery, as well as the frequency of CD8 T effector memory cells in the periphery. Finally, expression of cytotoxic and effector genes by these same T cells was also associated with improved overall survival. Other people have shown similar results. For example, in this paper by Kagamu and colleagues published in 2020, he showed that the concentration of blood T cells in patients with non-small cell lung cancer was correlated with improved overall as well as progression-free survival. In this particular case, the peripheral T cells that were found associated with survival were CD62 low, CD4 and CD8 T cells, as well as regulatory T cells. The authors were able to combine these immune features into a model that predicted clinical outcomes with high sensitivity and specificity. TakeoBio is working on clinical studies as well. For example, at SITC 2022, we presented a poster with the preliminary findings from our work with the Huntsman Cancer Institute, in which we showed that concentrations in the blood of regulatory cell populations, including TIGIT-positive Tregs, as well as CD161 positive CD4 T cells, are correlated with presentation of severe adverse events. To be specific, these cell populations were present in lower concentrations in patients with severe IRA versus patients who would not go on to present severe IRA. If you thought this was interesting, you'd like to learn more, please reach out to us at takeobio.org, at TakeoBio. You can also find a link to all the papers cited in this presentation in the description of this video, as well as a link to a webinar by Dr. Spitzer about his latest paper in Cell, and to our poster that we presented at SITC 2022. Thank you for your attention.